The topic for today is microbiota-induced IgA antibodies regulate fungal commensalism in the gut and are dysregulated in Crohn's disease. It's one of a number of studies that are focusing on the action of secretory IgA in controlling the effects of pathogenic forms of Candida. Amazing subject. Very excited about this. And we want to know how that becomes unbalanced in some forms of inflammatory bowel disease, specifically Crohn's disease. Dr. Gonshor.
Hello, Reed. Well, this is a good one, and we'll discuss why it's so good later on. But as you know, I say all the time that in each one of these sessions, we take a deep dive into recent research that brings to light objective scientific evidence. Remember, it's got to be objective and it's got to be scientific. So let's look more closely. I'm not going to say that whole thing again, but there are a number of recent articles that some highlighted in today's session that are shining a light on the importance of maintaining a healthy level of the antibody SIG-A, secretory IgA. And what happens when fungi turn from these wonderful commensals to aggressive pathogens. So let's look at the actual evidence because science matters.
Well, thanks, Dr. Gonshor. This is an amazing topic. And of course, in FDN land, we pay a lot of attention to secretory IgA. We love to measure it. Mostly in saliva, of course. We find this a very convenient and inexpensive and easy way to get an accurate level of how the immune system is responding to things in the gut, like the fungus or fungi, as you mentioned. And I found it to be a fascinating study. But before we get to that, in our discussion last week of glucagon-like peptide 1 or GLP-1 and its role in enhancing insulin secretion, reducing appetite, weight control, we received several questions. So the first one, most popular question, I guess, was that you mentioned the meteoric rise in sales of drugs such as Ozempic and Wegovy, which were developed as anti-diabetic drugs. And apart from rare cases of paresis, the question was, what other negative effects of these drugs should we be aware of?
Well, I have to tell you that we're going to spend a few minutes more than we normally do because this is, to say the least, not a hot topic. It's a burning topic. It has taken over the world's conversation, and for good reason. So there are going to be two questions we're going to answer, and then there's going to be a comment. The most common side effects of these semaglutides, Ozempic, Wegovy, are nausea, stomach pain, constipation, diarrhea, and vomiting. So you can have that whole slew of problems that can occur. However, apart from the relatively rare occurrence of what you just mentioned, which is gastroparesis, stomach paralysis, and if you remember, it was the caveat that we talked about at the end of the last broadcast, there are a number of serious side effects, as there would be with most drugs. And these include inflammation of the pancreas, which can be quite intense and very worrisome. You can also suddenly get a tremendous dip in blood sugar levels. You can get very aggressive hypoglycemia, and you can actually get a number of kidney problems, which can lead to frank kidney failure. And there are also gallbladder issues, amongst others. So just remember, like any kind of drug, you've got to be careful. You've got to know what these things can do. As well, you need to remember that Ozempic was developed, as was Wegovy, as an anti-diabetic drug by Novo Nordisk. It is not FDA approved for weight loss. Wegovy is. So for the people who are really saying, where's that Ozempic? I'd like to utilize it. Remember, it's off-label, and it has to be done with certified people who know how to deal with these medications. So those who are using the drugs off-label need to be cognizant of these possible problems, and there are a good number of them. The second question, which I'll just read out, how do the newest medications like Ozempic and Wegovy, how do they work on obesity to help people lose weight? And it is a major paradigm shift. So I want to spend a little bit of time on this because you've really got to understand this. The reasons that we use medications to treat obesity is because obesity is now being very well understood as a disease. So like any other chronic and treatable disease, you need to target the disease pathophysiology and the mechanisms that lead to that. Now, remember, you can also do it without medications, but here we're talking about these two medications. And that's exactly what these medications do. They're targeting the pathophysiology. And most of them, interestingly enough, work in the brain. That's what is extremely important to know. For example, they do this by increasing satiety. That means you feel like you're full. You don't have to eat anymore. So it's critical because the brain therefore sets how much energy we want to store. It stores that energy in the form of fat. So our brain decides how much fat should we carry. And that has been known for a long time by a phrase called the defended fat mass set point. It sets the point for the amount of fat that the body is going to keep in its system. Basically, these medications help to regulate that set point and bring it to a healthier place. So what's happened in our modern Western society is that we have entered what is called the obesogenic environment. We're all getting obese due to what? Due to factors such as highly processed foods, lack of sleep, a lot of stress. Guess what, guys? We can deal with a lot of these issues, which won't have to have us utilize these medications. And I know, Reed, you stress that all the time. Excuse the pun of stress. Mm-hmm. And all of this contributes to this obesogenic environment. So what it's done is it's pushed up that defended fat mass set point. We're all sitting at this higher set point, which is why so many of us carry extra weight and specifically more fat. So these new medications help to do what? They decrease that defended fat mass set point, which in turn helps us carry a healthier amount of fat. It brings it down. So therefore, those taking the latest medications, what do? They feel like they don't need to eat more, and they feel full sooner. They find themselves eating less food. Overall, it's an experience of not wanting or needing to eat as much, and yet feeling quite satisfied with what one is eating. And during that process, because you're not eating as much, you're losing weight. Again, the brain on those taking the meds wants them to carry less fat. And so they eat less. And eventually those individuals lose weight. So they're able to defend a new fat mass set point, which is lower. So that's a lot of words, but it's telling you that there's really something real going on here. There's a paradigm shift. Those are the two questions. However, as I said, we're spending a few more minutes because just hot off the press is a major study from the New England Journal of Medicine. This is a top-notch journal, and it's talking about semaglutides, that's like Ozempic and Wegovy and some others, in patients with heart failure with preserved ejection fraction and obesity. I'll explain that. In this major peer-reviewed study, the authors concluded that in patients with heart failure and obesity, if you treat them with one of these semaglutides, it led to a larger reduction in the symptoms and physical limitations. They're able to do more exercise. They have greater improvements in exercise function at the same time with a greater weight loss than the placebo. So these findings came from a study with over 529 randomized patients. And it has immediately propelled this weight loss formulation of semaglutide into the ranks of agents that are used to prevent cardiovascular disease as well. This is big. And in the words of one of the lead authors, it's going to trigger, quote, a paradigm shift for cardiologists who will now need to consider prescribing this weight loss medication to many of their patients, agents that until now were not part of the usual pharmacological toolbox for cardiologists. So we're going to provide this New England Journal study on the website for those who want to read it. Now, remember, we're talking about medications. Let me be very clear. There's a lot that can be done without having to go to those medications. Let's not forget that. There's a lot that can be done in lifestyle management and any number of other things that can decrease this massive problem of obesity and diabetes, etc. But for those who end up on the medications, it's turning out to be a really important series of GLP-1s. So that's all I'm going to say on that basis, but it's very important.
A good one. And I think there's a theme that I'm noticing develop. This is only our 8th or 9th Science Matters episode. And I think it's amazing that we can present both the pharmacological or medical point of view. A lot of the studies are based on that kind of investigation. And there is the practical application. When people are really sick, they have a true medical condition, it's always good to go and get checked out by a medical doctor who has all these wonderful tools and agents at their disposal. So they could help someone who's in really bad shape get out of the woods, so to speak. And I love what you just said about how the studies have indicated that after a while, you start feeling like doing more exercise. You probably would pay more attention to what you're eating,
exactly,
which is what we wish you did in the first place. And what we're there for is to help you figure out the diet and the rest. You mentioned sleep, the exercise, of course, is critical. The stress reduction is so vast, so many different ways, so many different types of stress. And of course, we like supplementation as natural as possible. But again, I've never in all of my training of people said that relief care isn't important. And so if you can intercede with some of these things and get the person paying attention, save their life, so to speak, then they're much more able and probably willing, we hope, to get into the lifestyle medicine that we teach. And so I want to thank you for that and for allowing for a discussion of both sides of the equation here. And so, because there's no FDN that really writes prescriptions, I just wanted to get that out there.
Yeah.
Our prescription has always been lifestyle. And yet some people are just, especially these people with the heart failure and extreme obesity, they need something to get them out of the woods. A good friend of ours says, get the life preserver on them, while they're drowning in this pool of poor health and get them over to the edge. Well, then we can step in and take, if they're willing, to do some of the lifestyle medicine things. So I think it's a great combination. And I want to thank everybody for submitting your questions. This is how we learn. They're very important. And with that, Dr. Gonshor, let's jump into today's presentation about microbiota-induced IgA antibodies and how they regulate fungal commensalism in the gut and are really, really important.
Well, thank you, everyone. And today, as we said, is that we're going to be discussing microbiota-induced IgA antibodies and how they regulate fungal commensalism in the gut and are dysregulated in Crohn's disease. So what are we talking about here? Well, I have included in this case 2 studies. The one on the left is the one that really we're going to be discussing, but I've added another one because it deals with something which is going to be very important in understanding this study that we're discussing. And this one, the second one, says that IgA, this antibody, targets hyphal forms of Candida. And we're going to discuss that in a moment. And its cell surface adhesins that exacerbate intestinal damage. So hyphal forms are the ones that exacerbate intestinal damage. And the immune system seems to be able to distinguish between benign and harmful Candida forms. Keep that in mind because we're going to go through that in a lot of detail. And the study that we're going through today says that antifungal SIGA, it's a portion of the total SIGA population, which is produced in the gut, can play a role in regulating intestinal fungal commensalism by coating fungal morphotypes that are linked to virulence. In other words, that this SIGA is able to really attack those types of candida which are virulent, and in that way providing a protective mechanism that for some reason seems to be or might be dysregulated in patients with Crohn's disease. So before we go into the study itself, a quick primer on how SIGA, what is SIGA and how is it formed? And those who've taken some of my other lectures know about this. If you take a look at the layer of the lamina propria, which lies right up against the epithelial layer in the gut, the one cell layer, you know that there are plasma cells there. And these plasma cells in the submucosa secrete a polymeric IgA. And they're united by a little protein, which is called the J protein. And so these plasma cells are producing these IgA antibodies. And these antibodies come up against the intestinal cell and they join with a polymeric IgA receptor at the surface. It's that series of little brown chains that you see here. And that whole kit and caboodle enters into little vesicles in the intestinal cells. They move through the intestinal cell. And while they're doing that, a portion of this particular receptor and the polymeric IgA gets cleaved just before it enters the lumen. So what you end up with in the lumen is the secretory IgA, which is made up of these IgA polymerics together with this little component of the receptor, and that is what's called secretory IgA. So that's what diffuses into the mucus of the lumen. So in the mucus secretion, there is IgA that has an additional secretory component which is created in these cells. And what are the functions of this secretory IgA? Well, we know them as immune exclusion. What does that mean? SIGA has an incredibly important function. It's the first line of defense in infection resistance. Before anything else happens in the body, in the gut itself, SIGA seeks out what may be causing infection and gets to them. And so it prevents microbial pathogens and various antigens such as toxins from gaining access to the rest of the body, and it creates adhesion to intestinal cells. So before it can adhere to intestinal cells, what does it do? It has a line of defense which is made up of three major things: agglutination, mucus entrapment, and peristaltic clearance. Well, what's agglutination? Well, we know that that's cross-linking. You cross-link any invading microbes or toxins, and that way you bind them. And if you bind up with them, you neutralize these exotoxins and various microbes. So that's one of the things that SIGA does. The second thing it does is there's mucus entrapment. This SIGA sits in mucus. That's one of the reasons you need a mucoid layer in the intestine. And these mucus entrapment of the microbes, which is where SIGA lives, it inhibits the ability of these microbes to actually move around. It literally holds them in place in the mucus. And when it's got them held in place, it then clears them out of the gut by just moving, by muscle peristaltic movement. It gets them out of the gut into the feces before it can get into the body. So that's SIGA. The other issue that we need to make sure we understand, and is going to be discussed in this article, is Candida. Well, what about Candida? Well, Candida albicans actually exists in three forms. It's important to know that. It exists as a commensal form, the classic commensal yeast form, and it sits as a commensal with all the other microbes in the gut and really doesn't do much damage unless you're immunocompromised and for whatever reason are not keeping it in check. But normally it's commensal. But you can also get a movement towards a hyphal form. And when you get a hyphal form, that's the aggressive pathogenic type of candida. It can invade the host tissues. It can breach endothelial cells. It can lyse macrophages, neutrophils, etc. And that's what you want to be aware of, is when does it become a hyphal type of Candida albicans? And so in this study, they looked at the various ways in which the cells and other aspects in the submucosa of the gut lining, what does it do? And we know that there are beta cells, there are dendritic cells, there are all kinds of macrophages, etc., which normally will send out the SIG-A, it'll find out what's going on, and it'll be able to send all of these different cells both into the luminal area, and if anything gets through into the other side of the endothelial cells, it's going to fight them and make sure that these toxins and microbiota don't get any further. That's not what happens when a human has Crohn's disease, or when you create in mice models the same thing as Crohn's disease. What happens then is you destroy the numbers of beta cells, you destroy a lot of the dendritic cells and so on, and you cannot produce enough SIG-A to be able to fight the various toxins, etc. And that's what happens in Crohn's disease. So SIG-A is involved in maintaining specific intestinal fungi, such as Candida albicans, in its benign form. SIG-A is able to figure out what it needs to do not to allow the hyphal form to start to increase in numbers. But in people with Crohn's disease, this process of keeping down the amount of the hyphal form gets interrupted. So again, SIG-A selectively binds to the form of Candida, which is hyphal, that is pathogenic, and it stops its spread. Quite a neat trick, by the way. So SIG-A binds to the sites on these hyphal forms of Candida that produce various molecules that are used by these fungi to harm host tissues. If these pathological forms of Candida want to get through the tissues, they need to have little molecules that can literally get them through these various barriers. So antibodies like SIG-A may help the body maintain a healthy balance of gut fungi. And how does it do it? By preventing the harmful forms of the fungi from taking over. That's a neat trick. So remember, SIG-A binds microbiota, various bacteria, fungi, and in that way modulates the intestinal immunity and homeostasis. It cross-links the various bacteria and fungi in the lumen to prevent them from encroaching onto the intestinal epithelium. Remember, in the lumen, the system does everything it can, including having a good mucoid layer, etc., to not allow any of the toxins and pathogenic bacteria and fungi from getting close to the actual cells themselves, because if they do, that causes some real problems. So the result of all of that is that we now know that there's a portion of these microbiota-driven SIG-A. This response is induced by and directed towards the intestinal fungi. And SIG-A, which is secreted in the gut, helps to control, in all of this that I've said to you, the pathogenesis of Candida albicans in healthy individuals. It keeps it down to a minimum. This protective mechanism, it seems, may be disabled in people with Crohn's disease. So what are the key takeaways? Antifungal SIG-A, which remember is a subset of the total SIGA. It's produced in the gut, and it helps to regulate intestinal fungal commensalism by doing what? By coating the fungal morphotypes that are linked to virulence, that is the hyphal form, and therefore, in that way, providing a protective mechanism that might be dysregulated in patients with Crohn's disease. And when used therapeutically, it might be a way to help Crohn's patients who develop an overgrowth of C. albicans. And remember, not all patients with IBD, that's inflammatory bowel disease, have this type of fungal overgrowth, but it may be an important contributor to disease in a subset of patients. And with that, thank you very much. Can't hear you, Reed.
Thank you, sir. I said, oh boy, I could just hear the questions coming in now, Dr. Gonshor, because this is such a fascinating area. We've studied secretory IgA extensively. We test it in just about every single one of our clients. We want to know their secretory IgA levels. And now we know that it's not just fighting bacteria, perhaps, if it's elevated, that it can be fungus. It can be all these toxins. Again, you talked about the cross-linking, which I think is really a critical consideration, and gives us insights how we might go about some healing, except for, in the cases of Crohn's disease, which makes it even worse. That's more of a medical condition, and we want to give a lot of special attention there. So thank you again for that. I want everybody to subscribe, like, and share if you enjoyed this episode, and submit your questions and comments. I know there'll be lots of them on this episode. Oh, and by the way, any suggested topics, please submit those things to fdntraining.com/science matters. And to grab a copy of the studies that we've been doing, there's now quite a archive building up at fdntraining.com/studies. And join us again in 2 weeks on October 18th, when we talk about the mechanistic link between gastric cancer and H. pylori. Remember, now that's another fantastic area, because we test people for H. pylori, and there's some things about the virulence factors that you're going to learn from the next episode, because